In this video, we will discuss about safe immunosuppression resistance Glycan dependent pan cancer immunotherapy. So this is a kind of approach where it will have a more specific attack on the cancer cells, whereas less or non-specific attacks on the healthy cells. So, and this strategy focuses on the glycans to detect the TACA, that is tumor associated cancer antigen. And so far the problem with the many immunotherapies is that they are not able to overcome the immunosuppressions or the tumor microenvironment and not able to reach to the cancer sites. And they most of the times they also have a non-specific attack on the healthy cells. So this approach that is presented in this is the safe immunosuppression resistance glycan dependent pan cancer immunotherapy. Right now this is a preprint, but it has been there in the Cell journal. So we will discuss what this paper says or how they describe their safe immunosuppression cancer immunotherapy. So they define this glycan dependent T cells receptors as GlyTR, and they call them as safe cancer immunotherapeutic targeting via velcro like binding of multivalent lectin to high density tumor associated carbohydrate antigen. So it determines the high density TACA, so that even though normal cells express this TACA at low levels, it will not attack on these normal cells. So we will discuss the graphical abstract today. So this is the glycocalyx, which is usually we can say that collection of carbohydrate structures that are expressed on the cell surface, and the cancer associated or the cancer cells or the tumor cells express glycocalyx that have high density expressions of this TACA, this tumor associated carbohydrate antigens, such as in when it comes to the N-glycan, it is beta-1,6 branching. When it comes to the O-glycan, it is TN antigen, STN antigen, and some of the others TACAs are GD2, GM2, LAC-DNAC. And their approach is that their T receptors or the CAR T receptors that contains this GlyTR1 or GlyTR2, it will attack on the cells which has this high density expression of TACA, not on the cells which has the low density expression of TACA. This is how they design their uh, T cells or glycan dependent T cell receptors. And one of the other advantage that they have shown is that it can overcome the immunosuppression's tumor microenvironment, that is the T regulatory cells or the immune checkpoint inhibitors by these PD-1 interactions that will be inhibited on these T cell modified T cell receptors, glycan dependent T cell receptors, and it can continue to attack on the cancer cells and even overcome these hypoxia conditions, TGF beta, interleukin 10, and other immunosuppressive conditions. And when they check their GlyTR on the diverse cancer in the mouse model, they are able to see that in the case where GlyTR is used, the tumors is significantly suppressed compared to its control. And thereafter, they also check the patient samples to confirm that this GlyTR overcomes tumor microenvironment even with exhausted T cells. So they took the exhausted T cells, they took the fresh tumors and then check it for the exhausted T cells, and then makes the organoid, and then use this GlyTR CAR T cells in the exhausted T cells to check whether it is able to affect these tumors or not. And they are able to see that the live cells or tumor cells in the case of this GlyTR is significantly live tumor cells in the case of GlyTR is significantly lower compared to the control case where the tumor cells are still alive. So this overall the graphical abstracts indicates that their immunosuppressions might be one of the therapeutic approach in future to target the solid tumors that are being shielded by the tumor microenvironment and the other immunosuppressions factors as there, and at the same time the non-specific attack or the toxicities on the healthy cells using their CAR-T glycan dependent CAR-T receptors is very lower compared to the traditional CAR-T therapies, immunotherapies. We will discuss their results in our subsequent videos. I hope you understood their graphical abstract at least to get an idea of the paper. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.